Welcome everyone to the first lecture of Perceptions on DNA Nanotechnologies. The speaker for the lecture is Professor Amanda Ellis. Professor Amanda is the head of Department of Chemical Engineering at the University of Melbourne. She obtained her Ph.D. in Applied Chemistry from University of Technology, Sydney. Professor Amanda is a well-known researcher in DNA nanotechnology, novel polymer coatings, and plastic and biomass gasification. I'm sure the audience is eagerly waiting to hear you speak, Professor Amanda. Handing over to you, Professor.
All right. Thank you very much, Naranjan and Kriti, for inviting me here to give you a little introduction on DNA nanotechnologies and some of the things that we're doing at the University of Melbourne in the Chemical Engineering Department. I guess the structure of my talk is really going to be, I'll do an introduction about the University of Melbourne, and then a little bit of an introduction into DNA, because many of you might not know actually what it is, and it's very important to understand the nuances of the structure and how it can self-assemble, and then I'll give you some real-life examples and things that others have been doing and also we've been doing in chemical engineering. It's very new for chemical engineering. It's not really an obvious an obvious thing for chemical engineers to do, but it's certainly at the forefront of some future science, and so it will be something that you'll be using, I would hope, in the coming years in technology. About Melbourne at the University of Melbourne, and so this is in Australia, which is right down at the bottom of the planet, right next door to New Zealand, which is where I'm actually was born. So I'm actually from New Zealand, and so I've migrated to Australia. And Melbourne is at the bottom here in the state called Victoria, and it's the capital of Victoria. And this little island down here is called Tasmania. So we have a nice relationship with Tasmania, even though there's a lot of jokes that go about it not being connected to the mainland. Over the years, it's been thought of as one of the most livable cities in the world. I think it's the third most livable city in the world now. We have a small population, 4.4 million out of the say 25 million that live in Australia, but we have quite a diverse number of nationalities in Australia, with about 110 different nationalities, and of course India has a very large population of peoples in Australia, and certainly in Victoria. So the University of Melbourne in Australia is deemed the #1 university in Australia according to the Times Higher Education World University Rankings, and it's the 32nd in the world in terms of universities. So we're very proud of that. We work very hard. It's an old university. It's in the city. It's established in the city. So this part here is our university. And it's made up of a lot of what we call feeder schools. So these are very old traditional schools like Trinity College and Queen's College and things like that. And the main campus really is here. I hope everybody can see my pointer, my laser pointer. But anyway, this is it here. And then you've got the main city here. So we're very close to what's going on in the world. It's a lovely campus, and you'll see it's got lots of lovely green trees and everything. And we're very well connected with, when we look, you probably can't see, it's not highlighted, but there's a massive medical precinct down here, which is all the medical hospitals and the medical institutions, the cancer research institutions, and various other things here. So anything to do with the biomed sector, we're very connected to. In terms of engineering, it's established in 1856, which might seem quite young to India, but to Australia, we're only sort of like 250 years old. So it was the first university to be established in Australia in 1856. And this is the building here, so we're very proud of that. And our ranking in engineering and technology is about 28th in the world. The good thing about our degrees, and I'm not quite sure about your Indian chemical engineering degrees, but I'm sure they still are accredited. Ours are accredited by the Engineers Australia and also EU RACE and IChemE, which is the national body in the UK for accrediting chemical engineers. So we've got a lot of accreditations for our Chem-Eng degree. Our engineering students, we have around about 11,500. At the moment, most of them are doing IT, which is not surprising in this climate. So we have around about 420 chemical engineering students and around about 90 Ph.D. students in chemical engineering. And overall, around about, we have quite a high representation of females in engineering, which we're very proud of as well, because it's very often very difficult to get women into what we call STEM, so science, technology, engineering, and mathematics subjects. And again, we have a very diverse engineering cohort of around about 102 different subjects, students from all over the world. So our department itself is quite versatile. We do a lot of bioprocessing and food engineering, including pharmaceutical engineering. We do a lot with nanomedicine and biotechnology. So we have some quite famous academics, Frank Caruso and Sandra Kentish, that do lots of nanomedicine and biotechnology. We do materials design and multi-scale modeling. And also we do a lot more in terms of sustainable technologies and processing and remediation. And one of our researchers does remediation down in Antarctica, which is obviously in the South Pole. And because of human intervention down there, there was also a huge need to remediate the land now because of the damage that we've already been doing. So that's some really interesting work and projects that are down there. So other than that, we do the traditional wastewater management, we do membrane science, we do polymer science, we do nanotechnology, and I'm just going to sort of introduce the Dna Nanotechnologies, which is obviously not scalable at the moment, let's be honest, but it is something that's on the radar in terms of things that chemical engineers can do. And then we also at Melbourne have a broad range of all of the fabrication and characterization techniques that you require. And this is for biomaterials right the way through to materials to batteries, characterization, membrane characterization, etc., etc. So we have a lovely broad range of things there that we can have access to. So that gives rise to, exceptional research. As you'll know, either as undergraduates or as postdocs or PhD students, it's the infrastructure that's around you that gives you that ability to do some really interesting and exciting, innovative research. So, the more that your universities can provide that for you, then ultimately the more successful you're going to be as a researcher. Right, so I just want to briefly just talk about some of the research that I have done over the years, and not just, and then sort of at the moment I'm just going to talk about Dna Nanotech. So we've done a lot of work with bio and microfluidics. We've done a lot of work with biosensing and a thing called surface enhanced Raman scattering, where we can analyze single molecules on surfaces and various other types of biosensing, whether it's anions in solution or cations in solution, or bacteria in solution, viruses in solution, that sort of thing. We've also done some work with things called quantum dots and carbon dots, which are sort of tiny little particles that, because of quantum confinement, when you shine light on them, energy is increased, and then they emit that energy, and so you end up getting some sort of fluorescence coming out of the material. And you can do that with carbon, and you can also do that with heavy metals, so things like cadmium sulfide and cadmium selenide and things like that, just due to the quantum confinement that's going on. And then we've done a lot of work with graphene oxide, a lot of work with drug delivery and water treatment. So this is using things like alginates and various other biopolymers to sequester pollutants from the environment. And so that's always been very interesting, doing that sort of work. And I suspect that in India, you're still trying to remediate some of your water. And so things like using alginates and things like that as absorbers of textile dyes and things like that is still very much of interest in the chemical engineering field. And as is using polymer membranes. So we have done work with polymer membranes, and particularly looking at trying to stop the fouling, the biofouling that occurs on membranes, because unfortunately most water systems have a lot of microbial activity in them. So, you know, bugs and things like that floating around in the water. And when you try and pass that through a membrane, they get stuck on the membrane and then they start growing. And that then stops the filtration mechanism of the membrane. And so we look at trying to modify those surfaces so that the bugs can't stick properly, and we can, you know, get more lifetime out of the membranes. The work that we were doing with desalination, for example, the membranes might last 2 or 3 years, and then you have to throw the modules away and put them into landfill. And so that's catastrophic, really, for the environment. And because you might have millions of membrane modules that you've got to replace every 3 years around the country, around the world. So then finally, what I've been working on, I guess, a little bit as well, has been this Dna nanotechnology, which is quite novel and relatively new science. We all kind of understand a little bit about carbon nanotechnology, where you're looking at carbon nanotubes and graphene and carbon dots and things like that. But Dna nanotechnology is a lot more complex and a lot more expensive. I mean, there's no other way of describing it other than it being a very expensive activity to undertake Dna nanotechnology. So let's start now with an introduction to DNA. Does anybody, before I begin, have any questions they'd like to ask about the University of Melbourne or anything like that? I don't know, Krita, has there been any questions so far?
Yes, we actually received some questions about what the role chemical engineers could play about in the field. So I think you covered that to some extent. So I think those were most of the questions. I'll pause you again at around after the next 20 minutes to take up further questions. Is that okay?
Okay, all right, thanks, Kriti. Okay, yeah. So let's start with an introduction to DNA. Now, many of you probably have learned some of this in your undergraduate at some stage, maybe first year, if you've done any first year biology. I'm not quite sure how your undergraduate is structured, whether you just do chemistry and maths, obviously, and probably some physics, maybe. But I'll just give you a brief introduction to DNA. So the good thing about DNA is it's everywhere, right? And we know a lot about it. What we don't know so much about is how to manipulate it and how to make it on large scale. And this is what really DNA nanotechnology is aiming towards, is how do we manipulate it and how do we manufacture it on a larger scale? And that's where chemical engineering can, in the future, potentially come in. So for those that don't know much about DNA, we share about 50% of it with a banana. So you can see that that other 50% is quite important because there is a big difference between a human and a banana. Contain about 2 to 2.5 meters of DNA in each one of our cells. So all of the DNA in your body was put end to end. It would reach to the moon and back about 600 times. So we've got a lot of DNA in our body, as do other organisms on the planet. It represents about 99.9% of it is identical to each other. So it's only 0.1% that differs from each other. And this is, I'll talk a little bit about these mismatches that we have in DNA. And it's those mismatches in particular parts of our genome that give rise to, or mismatches, or the anomalies in our genome that give rise to different things like our eye color, our hair color, whether we, whether we can take particular types of medication or not, or whether we can roll our tongues or whether we can't roll our tongues. Those sorts of really crazy things that make us all individual from each other. And so 3 million nucleotides different, and I'll explain in a minute what a nucleotide is. You can store information on DNA, which is something that DNA nanotechnologists are very interested in doing. So basically you can store about 25 gigabytes of information per inch on your DNA. And if you look at, you can go to the moon 600 times back. That's a lot of information that you can store on all of our DNA, which makes us, much more intelligent than computers. But we have to harness that. We have to learn how to understand how to harness that kind of ability to use that information generation and storage. The important thing here is that we eat a lot of DNA. So DNA really is harmless to us. It's not, if you're manipulating it in the lab and you're making it or synthesizing it in the lab, there's no danger to that. You're not going to be modifying your genome or doing anything crazy like that, because we do eat a lot of it, and so it's pretty harmless. Again, if you were to be a person and you were going to typing at 60 words per minute, 8 hours a day, it'd take you around 50 years to type the human genome. And yet we can sequence the human genome now very rapidly in about 20 or 30 minutes, and that will give you your entire genome. But nobody has a clue what that means, right? So you can give your whole genome to somebody and nobody's going to know what it means, because we haven't worked out yet what the sequence actually represents. We know what it represents at very specific regions, which might be attributed to a particular disease, but we don't really know what the whole one is. And so, just while you probably listen to the minute, your body will reproduce enough new DNA that could stretch 100,000 kilometers. So our body's really good at producing it on scale. Our biggest problem is how do we produce it out of our body and synthesize it so that we can use it for different applications. So that's our biggest issue. So just in terms of how it was discovered, I'll go through this really quickly. So there was this guy, Frederick Griffin. He introduced this transforming principle, where basically he took two bacteria. One of them was harmless to mice, and one of them killed the mice. He injected them in. The first thing, the non-virulent strain, the mouse lived, and then the other one died. Then what they did is they took the bacteria that was the killer and they heated it up, and then they re-injected it again. And then this time the mouse lived. And so what it meant was that you could kill the bacteria by heating it. But then when they put that heat-treated material with the one that usually lives, what they found was the mouse died. So what that meant is that while heat treating killed the bacteria, it didn't kill the DNA. And in fact, what happened was that this non-virulent strain uptook the DNA that was dangerous and then infected the mouse. So that was what they call the transforming principle, where this DNA could go from one type of cell into another cell and still be aggressive. And they didn't know, they had to then find out what that material was. So they didn't know what the material was. It was discovered by Oswald Avery in 1943 that digested it all up into tiny little pieces using these particular enzymes that can chop up DNA. And then previous to that, this man here, Phobos Levin, in 1929, had assigned this material a specific, not a structure, because the structure wasn't worked out then at this stage. It was just what was it composed of. So he decided these things were going to be called nucleic acids, and that they were going to be composed, they were polymers, and they were going to be composed of nitrogenous bases, which are purines and pyrimidines, and I'll tell you what those mean in a minute, a sugar and a phosphate. So you've got these three components here. So there's the base up here, and then a sugar, and then a phosphate. So the two of them, a base and a sugar, is termed a nucleoside. So this is a nucleoside when you've got just the base and the sugar. A nucleotide has all three, so a base, a sugar, and a phosphate. And that is the principle, that's the building block, that's the monomer of the DNA polymer. And it's very important that you understand that you must have this phosphate group here for it really and truly to be a DNA analog. A lot of people do research on the base and the sugar and on nucleosides, and they put them into, they put them onto polymers, and they put them on nanoparticles and things like that. But that's not actually the true, it doesn't give rise to the true properties of DNA. You need to have that phosphate group there that make the nucleotide. And so that, I can't overemphasize that. So the sugars that are on your nucleotide can be a ribose. So this is where you have two hydroxyls here. So the important part here to notice is that you have the four-membered ring here with another fifth carbon here. And this is how DNA is numbered. So it's number 1 on the sugar, 2, 3, 4, and 5. And if you have two hydroxyls here, then you have ribose, so it's RNA, which we're not going to talk about very much. And if you have hydroxyl missing here on the 2, so it's 2-deoxy, so you've removed the oxygen, ribose, this then is your DNA sugar. So this is your DNA sugar here. And you'll notice 1, 2, 3, 4, and 5, right? And then you've got the bases. So the bases are equally as important. You've got the pyrimidines, which are single aromatic rings, and you have 3 of those. So uracil is only for RNA. You don't have that in DNA, uracil. So DNA contains cytosine and thymine, which are the pyrimidines, and then it has the adenine and guanine, which have the 2 aromatic rings, and these are the purines. And how these guys get together is absolutely implicit in DNA and DNA nanotechnology, right? So how these things get together. So this guy here, Erwin Cargill, he came up with a rule that said that for every A that you have the same amount of T, and every G you have the same amount of C. It's changed over time, but this is just the fundamental rule. And all it basically means is that A only binds to T and G only binds to C normally in a normal canonical binding situation. All right. So when you look at it here, this is DNA effectively. So you've got the guanine, which is your pyrimidine, and you've got a cytosine here, which is your, no, sorry, that's the wrong way around. This is the, I always get this around the wrong way. This is the purine, and this is the pyrimidine. And you'll notice it's got 3 hydrogen bonds. What that means is that's a stronger bonding scenario than if you have your adenine, which is your purine, and then your pyrimidine, which is your thymine, and that has 2 hydrogen bonds. The other thing to look at is now we have the phosphate, which is really important. That's on the 5' end. So down here is the 5', you know, this 5th carbon here. And you have a hydroxyl here on your 3rd carbon. DNA always goes anti-parallel to itself. So on one side we have 5' to 3', and then it links up with the other strand of DNA so that you have 5' to 3'. And that's very important that you have that situation here. And that's just, I won't go into the synthesis of DNA, but that's just very important. All right. Now, the important thing to understand is the bonding. So especially when you're looking at DNA nanotechnology, because you're talking about assembling these things based on this base pairing. So A and T always go together, which is the 2 hydrogen bonds, and G and C go together with the 3 hydrogen bonding, and that's called Watson and Crick bonding. But you also have this weird other bonding, which makes up about 1% in our bodies, and can give rise to some unusual characteristics that we have. This is the, excuse me, the 0.1% difference that we have. And this is the Hoogstein, where we have mismatches. So you might have an A with a C or a G with a T, etc. And the DNA has to compensate by changing, it expands slightly or it rotates slightly just at that particular mismatch in order to try and get that into its structure. And that can cause that causes some diversity in our genome that gives rise to a whole broad range of things. But it's also very useful, these mismatches are very useful in DNA self-assembly, because we can use these as the dehybridization. So hybridization is when you get one strand and another strand come together. Dehybridization is when you pull them apart. And when they go together, they do that at a particular temperature, and when they're pulled apart, then they can do that at a different temperature. And we can modulate those temperatures quite easily by modulating how our nucleotides bind together, how our bases bind together. So, of course, most of you should know that Rosalind Franklin was the postdoc of Watson and Crick, and she did the X-ray diffraction of DNA and discovered its structure. And then they got the Nobel Prize for it, and later she's been acknowledged also as being part of that Nobel Prize. So that was good news for women power. Then in terms of the physical structure, one of the things now we know that when this gets together, you've got the 5' and the 3', and you've got all of this hydrogen bonding in here, it gets so constrained that it has to twist. So it twists into a helix, and that's our DNA structure. And in that DNA structure, you've got what we call major grooves and minor grooves. The distance between each twist is about 3.5 nanometers, and the distance between each of the base pairs that are in there is about 0.34 nanometers. Now, what we can do is we can put dyes inside the structure, and that will make us, we can look at them, that way we can look at them, and we can also put dyes in the major groove and minor groove. So often you see people talking about different dyes that, you know, intercalate into DNA using the major groove or the minor groove. I won't go to too much. So single-strand DNA is less stable than double-strand. The double-strand DNA has a persistence, so that means it's sort of a rod-like for about 3.9 nanometers, and then it's floppy. Any base pair that is not canonical, so not Watson and Crick, is called a mismatch. And we can change double-stranded DNA by modulating the different temperatures. So if you put in an A or a T, you increase the melting temperature by 2 degrees. And if you put in a G or C, you increase it by about 3 degrees. This is because this has the 3 hydrogen bonding and this has the 2 hydrogen bonding. So if we look at temperatures, we can see here we've got something like a poly Da and a poly Dt. So that's just an oligonucleotide of A and an oligonucleotide of T. So they're completely complementary. As we increase the strand length, so we go from 5 nucleotides all the way up to 20, we increase the melting point. So that's because we've got more bonding. But if you look at the blue, is the AT is much lower melting temperature than the higher DG, DC, which has a higher melting temperature. So we can play around with that when we're trying to build our DNA structures. So knowing what DNA is and how can we what we know about it now, what is it that we can do at the nanoscale? So are there any questions now at this stage?
Yes, Professor. So I think we have the first question, which is from the previous part, which says, can we reuse the membranes in any manner as their waste leads to harmful environmental effects?
Can we use the membranes?
I think this was for the previous section, the one before you talked about the DNA. So it talks
I'm not
about
quite
the
sure.
reuse of membranes. I think it was regarding the specific case wherein you pointed about the purification of water. I think it was about that.
Yeah. Well, I mean, there's many different types of membranes, and it depends on the dirtiness of the water. Often there's always pre-treatment prior to a membrane, so you might use a sand bed or something like that, or some sort of nanoparticles or flocculant prior to it going onto a membrane. But the membrane will get rid of any, if it's nanofiltration, you'll get rid of viruses and bacteria. If it's microfiltration, you'll just get rid of microparticles and things like that. So it depends on what your application is, really.
So the next question that we have was, do Hoogstein nucleotides indicate mutations?
It depends. It depends if they are different, if it's not supposed to be in your genome. So it's very difficult to know at this stage. I mean, usually they would call them single nucleotide polymorphs, but what that means is that if you have a different DNA sequence or mismatch compared to your parents or something like that, then that would be deemed as a mutation. If you have the same as your parent, then it's not a mutation, if you know what I mean.
Thank you so much. So the next question was, what are the non-canonical bonds for DNA?
So the non-canonical bonds for DNA are the Hoogstein bonds. So they are anything that isn't AT or GC, which is Watson and Crick, because they, designed the structure of DNA, which was 100% DNA without any mismatches. Anything else is a mismatch, and it's termed a Hoogstein bond. Yep. That's non-canonical.
So I think that's about all. So you can continue.
All right, so let's get on to some of the DNA at the nanoscale. So in a sense, DNA has been around for a little bit since 1953, and Watson Crick discovered it. So we have this guy Sanger here, who basically started to put DNA and discovered that you had these nucleotides, and then along came this guy and started putting these nucleotides onto microarrays, and then trying to put the complementary nucleotides onto them. And then the big breakthrough really came from this guy here, Mullis, who designed polymerized chain reaction, which is the amplification of DNA, or the production of DNA outside of the human body, where we use these NTPs, what they call the nucleotide, phosphoramidites, to couple together in a chemical reaction to create DNA. Then came along this guy Jeffries, who started to use DNA in fingerprinting. So that's when we started looking at human identification using what they call short tandem repeats. So every single human being has a different sequence at a particular point in your genome. So the long strand of DNA, billions of nucleotides long, at a particular point there, you will have a different series of sequences of your nucleotides than any other person. So you might have 5 GGGGGG, TTT, AAA, C, right? And that will be particularly distinctive to you. And so when we can amplify them using the PCR method, we amplify these sequences, and then we can identify you as being the person that's been responsible for a crime or something like that. So this is when DNA fingerprinting came out, only in 1984. So all of the crimes that were carried out way before then, and keep it well, where people are starting now to get charged with crimes that they committed before 1984, because we can now do this DNA sequencing. So it's been quite a miraculous discovery for humanity, really, and to try and help with crime. Now, since this time, this guy called Ned Seaman, who is the one that really uses computer programs to design DNA structures and then self-assemble them into DNA nanotechnology into different structures. And of course, the other thing that's come out is now we have the human genome sequencing. So we can, I can take my DNA, I can sequence the entire thing, and yes, I would know that I have 1 billion nucleotides, and they would be A, C, D, G, I, 5, but no one knows what that means at the moment. So nobody knows if I've got an A, a T, and a T, and a T, and a T, and an A together somewhere on my genome, that's going to mean that I'm going to have cancer or something like that. Nobody knows that yet. Anyway, so Seaman discovered these little guys. So he sort of developed the whole idea that we could take DNA, we could use mathematical modeling and different modeling softwares, and we could start to design these structures. Now, these structures are made out of 2 types of technologies. So one of them is the structural DNA nanotechnology, and the other one is what we call dynamic DNA technology. So we have the structural is the box, so that's is the box, and the dynamic part of it is the opening and the closing of the box. So how do we do that? And how do we can build the box, that's fairly straightforward, but how do we get the box to open and close? And that's the dynamic DNA technology, and I'll talk a bit about that. So we're looking at, you know, very small dimensions. And if you see here, this is an AFM image of little boxes. This is the box when it's closed. And because when it's closed, you can see some fluorescence. So when it's closed, we have a fluorophore here and we have a quencher. So when these two things come together, then you get no fluorescence. It's a fluorescent and a quencher quenches the fluorescence of the system. But because it's just dynamic, you're not going to ever get 100% of it being closed at any one time, because it fluctuates just because of Brownian motion and all sorts of other things that are going on. So you can see a little bit of fluorescence here. So it's not an AFM, it must be a confocal microscopy. So then you can see here, this is the box with the lid open, and when the lid's open, bang, all the little boxes light up, because now you have the quencher and the fluorophore apart from each other. And so you can start to see you know, something happening. So what you would do is you put a drug inside here, or some other material inside here. You can put this into your body because obviously DNA is harmless to human beings. It can float around. It can then release or be triggered by a different material inside your body, open up and release a drug into its local environment, into the cell, and maybe as a cancer-killing agent or something like that. So that's the whole premise of the whole thing. What else can we do with nanotech? Well, we can do the box open and closing. We can do nanomachines. So we might have a system where it's has a particular function, and then we put a strand of DNA in there, and that stops that function from happening. We can put in another strand, and then it starts that function again. So this all comes down into, in particular, these DNA logic devices, where, you know, when we have a machine, when it's on, you get a light, so that can be a 0. Sorry, that could be a 1, and when it's switched off, it can be a 0. So you can start to have 1 0 0 1, and you can start to build up computer logic devices on surfaces. Doing this kind of thing. So a lot of people do this around the world, making different DNA logic devices. You can also use structures, DNA structures, which I'll talk about soon, as that can open and close. So in one position, they're folded onto each other, they're all bonded together in a nice little ball. And then if you add something like an acid or a cation or something, they'll open up. And again, you've got the signal here. So this is a fluorophore and a quencher. When they're together, no fluorescence. When you put in some kind of analyte that you're looking for, then it opens up, and you get, and the fluorescence comes back on again. So it's quite easy to see what you're biosensing. And you can do things like protein assembly, and you can also do things like organic synthesis. Not so much for chemical engineers, but you could imagine if you had an enormous batch reactor and you had all of this DNA in there that you'd managed to synthesize or extract from plant products or something like that, because you could use that DNA. You can modify it with one chemical agent on one side and another chemical agent on another side, and when they bound together on the DNA template, then they'd undergo a chemical reaction, and then you would be able to get a product out of your batch reactor system on a large scale. So in terms of, and this is precise control, because you're really then doing a one-on-one reaction. So you're getting 100% yield instead of perhaps 75% yield in some of these chemical reactions that we do in reactors in chemical synthesis. So now I'll just talk a little bit about structural DNA. I don't know how much I'm doing for time. I think I've got another 20 minutes, I think, or so. So we're going to talk about these 4 structures, not necessarily D, but we'll definitely talk about these 3 here. which are the G quadruplex, the I motif, and the molecular beacon. And there is this other guy here, the thrombine, but we won't talk about that. So the simplest one is this hairpin loop. So this is the basic structure. So we have a DNA. Again, it maybe has a fluorophore and a quencher in each end, and that self-assembles to form this loop structure here. And you'll notice at the top there, that's called the stem, and that stem can be modified by changing those different nucleotides, and so we can have it open up really quickly if we want to change the melting temperature, or we can open it up really slowly by making it longer. So if you increase the length, you increase the energy stability of it, etc. If you want to put in mismatches, then you make it melt quicker, and all of those sorts of things. And this again is the stem is the Watson and Crick bonding. So that's the canonical bonding. We've done some work where you can change, rather than changing your nucleotides, you can put in dyes, different dyes, intercalating dyes into the system. So in this case, we've put in a dye called ethidium bromide. So you can see that there. It's a very toxic dye. It's very good at unraveling DNA, which is the last thing that you want to put into your body. Because so it's very dangerous, and they replace this dye now with things like CyberSafe and CyberGreen, which are a little bit more friendly. But the beautiful thing about this sort of dye is that it can speed up your assembly and disassembly process, which is required for DNA nanotechnology. And this is just an example of a fluorophore here. This is a fluorophore molecule, and this is a quencher molecule. And if you couple those together, then you get no fluorescence, and apart you get a fluorescence, as I said. So I won't go into too much into the G quadruplexes. What we've got here is, this is an RNA G quadruplex, and this is a DNA G quadruplex. And I think the most important thing to note about this is that we have these everywhere in our body. They're very important. They act as the ends of our DNA, of our genome, where they help with the telomeres, which stop our aging process or slow down our aging process. We get less of these G quadruplex as we get older, until finally we have none left. And basically, our DNA, just as it keeps amplifying, it just gets shorter and shorter and shorter. And then as it gets shorter and shorter and shorter, we end up getting more of these genes in our genome that get activated. So that might be a cancer gene, or it might be, you know, a Parkinson's disease gene or something like that. So as our genome gets shorter, we come across all of these different genes, and so eventually we don't last forever. But these are very important, and we have a lot of them when we're young, and we get less of them as we get old. And so what they are is, they are essentially oligonucleotides. So these are strands of DNA which have what they call G tracks. So these are three guanine nucleotides. And when you put them together with potassium, and that's why potassium is so important in our body, when you put them together with potassium, they will chelate around the potassium ion, and they will form all of these unique structures. They'll stack together. So this is one layer of the G quadruplex, and then you just get multiples of these stacking together. And this loop region N is the most important. This is the one here and down here that sort of gives rise to the different types of G quadruplexes that you can have, which are parallel, anti-parallel, and all of those sorts of things. Now, if you want to have a look at those, you can use a thing called circular dichromism, which is where we use polarized light. We have this in solution, and then you get this nice little spectrum here where you've got wavelength, and you've got ellipticity along the y-axis here. And the important thing to distinguish is, in this table here, is if you have parallel you've got these two different wavelengths, and it's the sign of that polarized light that's important. And so looking at this table, then you can identify the different types of structure that you've got. And they have all different types of activity, catalytic activity, and various other properties that you might need if you're trying to do use them as a biosensor or something like that. The other thing is this one here, which is called an eye motive. And an eye motive is also known as an intercalation motive. What you'll notice with an eye motive is it's a little bit different from a G quadruplex. G quadruplex is stack, so 1, 2, 3, 4, 5, whereas an eye motive crisscross. And so they're a little bit like a turnstile when you're going into the zoo or something. So it's sort of like it's a crisscross of only cytosine-rich sequence. So it's only C, and it will form in the presence of protons, not potassium, but in the presence of protons, it will form these structures, and it's only cytosine. And these little guys here are excellent for using them as pH sensors and things like that in devices. All right, now those eye motives are called Actimas, and basically Actimas recognize and bind target molecules. So they'll recognize hydrogen or protons, and they'll target those. Whereas a DNA zyme is more like a G quadruplex, and they have a particular catalytic activity. And so that's really important to distinguish between an Actima and a DNA zyme. So these are the things that are really more useful in, well, I mean, the eye motive is very useful, but a lot of research goes into G quadruplexes and trying to understand those. So for example, one of the things that we've done is we've used a G quadruplex. So you have the G quadruplex here. This orange is the potassium ion. And then when you use a cofactor, so you bind it with a thing called hemin, which is just this iron molecule that you can find in your blood. It's in your blood. And it's in the human body, so it binds well with these quadruplexes. You can then undergo different chemical reactions, and if you put them in the presence of a dye called ABTS, you can see the chemical reaction taking place, because you start to see an evolution of a blue color. And so you can tell, you can monitor your chemical reaction using these kinds of very complex systems. And what we did was we took a sequence which was G-rich. We coupled that with a antimicrobial agent, or a what's it called, antibiotic called oxacillin, and then we assembled it in potassium, so then it forms this structure here, and then we added hemin, which is our cofactor, and then when we put this into a biofilm, the oxacillin managed to penetrate into the cell walls of the microbes of the biofilm, and then the hemin and the DNA zyme underwent a chemical reaction and produced hydroxide ions, which then killed the bacteria in the biofilm. So it's got a lot of different applications in terms of being a transporter and also being a catalytic reactor, the DNA zymes. So ultimately what we can do is we can take these things and we can use them for detecting metal ions, for ion concentration. We can detect other DNA sequences, and we can use them also to suppress genes and disease treatment, and various other things using the DNA zyme, which is the quadruplex, G quadruplex systems. Now I want to get into the next thing, into more the structural part and the dynamic part of DNA nanotechnologies. So this is when we're trying to self-assemble. So we've made lots of DNA, and now what we want to do is we want to create, in this case down here, we've got a DNA sheet. Right, to do that, what we do is we take these sequences here. So we've got 1, 2, 3, 4 sequences. We put them together in a solution, and they form this thing called a holiday structure. And it's sort of like a knot, right? And then that knot binds to another knot, and then another knot, and then another knot, and then we end up having this sheet of DNA material. So once we've got one sheet, we can then start to combine that with another sheet, and another sheet, and another sheet, and another sheet, and another sheet. So we've got 6 sheets, and so we've got a box. We can get that to self-assemble into a box. And we can look at these sorts of things using microscopy. So we can use AFM, we can use TEM, transmission. So AFM is atomic force microscopy, TEM is transmission electron microscopy, and we can also do gel, native polyacrylamide gel electrophoresis, which is excellent at looking at different sizes of the boxes. And you can create all sorts of weird structures by this method, right? So using all these different holiday structures, we can use this computer program that's Seaman developed. So we can create all these weird shapes out of the sheets. And we can do this using this particular program here, which I'm not going to go into, but if you wanted to ever get into DNA nanotechnology, this is what you would use. And it's called, one of the examples is CAD Nano. And there's a lovely tutorial on how to use CAD Nano here. And you can create all sorts of structures, boxes, rods, cylinders, triangles, anything that you want, you can program into this device. And then what it will do is it will come up with a series of these things here, the DNA sequences, or what we call staple strands, which then can be used to create that structure. And it will self-assemble in solution based on the sequences that you have developed in your CAD Nano program. So it's quite complicated, but it's quite interesting as well. And so the typical reaction conditions that you use for these things is you take a bacterial DNA, and this is a circle. So if you imagine a rubber band, you take a rubber band, and what you're going to do is you're going to staple that rubber band at different positions, and then you're going to create a structure out of it. So out of that rubber band, you're going to create a box by stapling it at different points using other little pieces of DNA, and you're going to create the box. And it's the computer program that tells you how to do that based on the Watson and Crick bonding. So that's the, the A to T and the G to C. And based on that, you only need those 2 reactions, and you can create anything that you want using the computer program. And so you use different types of ratios of your staple strands. You have to have magnesium chloride in there because that helps with the bonding that is in DNA. That's why magnesium is also very good for human beings. You want to be able to keep your magnesium up and your potassium up to be as healthy as you possibly can be because DNA loves it. And we need it to manufacture DNA. And so those are the reaction conditions. The last thing I want to talk about, if I've got 5 or 10 minutes, is the DNA structural stuff. So this is what we call strand displacement reactions. So what you're essentially doing is you're taking a double strand of DNA, and what you're going to do is you're going to kick off that the red strand with another strand of DNA. And you're going to do that through this thing here, which is called the toe hold. And the toe hold is so this is single strand, that's about anywhere from 4 to 15 nucleotides long. So here we go, we've got a mismatched one, right? So we've got a little bit of a gap here. This thing will bond down, so it'll dock down, and you'll get the bonding, the hybridization. And then the next thing it will do, because it's thermodynamically more stable as the complete strand, it will kick the red strand off. Ding, 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 ding. So you get this base by base strand displacement. And so this is then thermally more stable. It's also very sensitive to mismatches and insertions. So if we have a match here, it will stop that reaction. It will just simply stop that reaction. So we isolated some human genome DNA, and we wanted to look at the male-female genome. So we wanted to distinguish between males and females using this technique. So we created this thing here, which has your toe hold. It has a fluorophore on the end. And then when we took and so basically the difference between males and females, males have 6 base pairs extra than the female. So they have a heavier gene here of 112 base pairs, whereas we have only 106 in females. And so these are really in what we call an insertion. So if you're going to do the displacement, you would expect that the reaction would stop here, right? It wouldn't completely displace. And that's what we see. So you go here, this is the quencher, this is the fluorophore. So we put the female in, and that completely quenches. We put the male in, and that stops. Right, and so what you end up is when you've got a perfect match, when it's a female genome gene, you end up with complete quenching of the fluorescence, and when it's not a complete match, when it's a male, then you get no, the fluorescence stays on. So it's an ideal way, in this way, of distinguishing between different genes that you've got there. So here you've got the if you've got a mixture of male and female. So it works perfectly well. And the interesting thing here is now you can start to put these sorts of things on microfluidic devices. So these are small channels, and you can put down what you're interested in looking at. You can hybridize the gene to it, and then you can dehybridize, and you can measure this quite easily in a microfluidic device. And so you can use this toe-hold strand displacement reaction or dynamic DNA reaction to go through. This is just some futuristic stuff, and it might be a little bit out of your pay grade. This is how you sequence DNA in your body. So you can see it's really complex. What we're trying to do is we're trying to sequence DNA outside the body, because at the moment you can only really sequence DNA up to about 200 base pairs. We want to sequence DNA up to hundreds of thousands of nucleotides long, which is unprecedented. So we want to do this through a sort of a templating mechanism where we do this horrible synthetic reactions to do that. And then we also want to do polymer DNA materials. So can you imagine if you've got a robot that's made out of a polymer, but we've integrated the nucleotides into that polymer so that then it can self-replicate. This is very futuristic, but this would be the idea, is that we create long polymer chains which have all these nucleotides on them, and then we can couple them to another polymer nuclear chain, and we can do self-replication of those organisms. That would be really a profound step forward in the next 50 years of science. So I'm going to finish there. I'd just like to acknowledge all the people that did the really hard work on this. So some of the postdocs and some of the PhD students that I've had that are now doctors and got their own academic positions around the world, and various funding and things like that I've got over the years. So I think I'll leave it there, Kriti, and thank you very much, and I welcome any questions.
Yes, Professor Amanda, we have a few questions with us. So I think you talked a few slides back about ethidium bromide. So can it be used for the purification of DNA?
No. Purification of DNA you would do on PAGE, on polyacrylamide gel electrophoresis or HPLC or something like that. It doesn't cleave the phosphate bond. It just disassembles the DNA. So it will make it go from double-stranded DNA, which is very integral to our body, to single-strand DNA. So you don't want that to happen in your body. So, but to purify DNA, to remove DNA, you'd need to cleave the phosphate, the phosphodiester bonds, which is the backbone of DNA.
Thank you. So the next question that we have is, are the DNA markers selective, or how does it work? Like, can we mark the DNA selectively?
Well, the selectivity arises from the fact that they will only bind, they'll preferably, that canonically, they will only bind to their complementary base pair. So that's their selectivity in a sense. So A with T and T with A and G with C. So that's the selectivity. I don't quite understand the question. Maybe they could rephrase it and send it back again.
Okay. So I think we can take another question in the meanwhile. So does the number of crossovers play any role in the stability of DNA structures?
Absolutely, yes. So the more crossovers, the more stable it will be, the more rigid it will be if you've got more crossovers. So some people want to make porous nanostructures so that are quite almost like chicken wire or something like that, mesh. And some people want to make very tight structures. So you then have to have a lot more crossovers there to make a more rigid structure. Because the porosity is also sometimes important in a nanostructure. But DNA is quite flexible, remember? So it's got a persistence length of about 3.9 nanometers. So once you get to a sort of a 20 nanometer structure, it's quite floppy. So you need to put scaffold, you need to put more connections in to stabilize it. It's like building a house. It's quite fun, actually. I mean, when you think about it, it's a little bit like trying to be an architect of a building. You're trying to design your own structure.
So the next question that we have is, there were recent studies that DNA can be changed to increase the life of a human, that is the lifespan. It also has the example of molecular scissors. So it said that since it is a process for just a single strand of DNA, and a human body has multiple, like infinite, we could take it to be infinite. So would you comment? Could you comment about it? How would that go on?
Well, nobody wants to modify the human genome in situ, for sure. Obviously, there's cloning and things like that have taken place on animals. The modification of our genome or DNA is not frowned upon. Is frowned upon. They do in particular with IVF. They can take out particular gene sequence. They can take out gene sequences, not put in new gene sequences, because that's not right. That's not allowed. There are animals in the world, in the kingdom, such as jellyfish, that don't age. And so there's a lot of research going into why that is. And it could potentially be because of their telomeres or these G quadruplexes that they might have, and that stop the unfolding or the unraveling of DNA. And so that can cause some longevity. I don't quite understand the question about molecular scissors, but certainly I'm not quite understanding. I don't know if they're talking about CRISPR or something like that, but I'm not quite sure. But I don't know what they mean by the molecular scissors.
Okay, thank you. That I really got to know a
Yeah, I'm
lot
sorry.
about it.
Didn't
Yeah.
quite. Yeah. So I mean, you can, with the persistence left, you can use DNA to pick up things and move things. It's very sticky. So if you were using AFM or something like that, you could potentially use them as a kind of tool. But I don't know, in terms of the molecular scissors, I'm not quite sure whether it's about CRISPR or what the question's about.
So, okay, we'll move on to the next question. So that is, I think it says CD nano programming. Is it the same which is referred to as DNA origami?
Yes, yeah. So it's basically to do DNA origami. Yep. I mean, structural DNA nanotechnology is essentially DNA origami. It's just, I didn't use that word. That's all.
Okay. So we have quite a few interesting questions. I hope you have the time to take that.
All right, as long as I can answer them, don't make them too hard.
Okay,
It is Sunday evening here, so
so I'll just probably take up just 5 more minutes of your time.
yeah.
So the next question is, what are some of the profound materials used in tissue regeneration, keeping sustainability into account?
Okay, so I think that's a different question than DNA nanotech sort of. I mean, obviously, you could look at the boxes and everything. I think you're probably looking more in tissue engineering. There's a lot of focus on obviously biodegradable polymers, right? And so things, but there's also a lot around now about nanocelluloses and things like that are becoming more interesting in terms of tissue engineering, and there's also a little bit of work around stabilizing tissues with reduced graphene oxide, things like that, which aren't biotoxic. So, yeah, I think that's about all I can come up with on the spot at the moment.
I think we have the last two questions that I'm going to take up. The first one is, can we modify the DNA of humans to regenerate organs like liver, which already has this type of ability?
Look, I think there's a big question. I think there's a big difference between can we and should we, and are we allowed? So at the moment, there is just no way that you're going to modify the human genome to do those kinds of things. I mean, more likely you're going to grow organs ex situ. So you're going to grow your kidney or something outside of the body and then implant it, but you're not going to modify your genome so that you can, get a better liver or a better heart or something like that as you go through life. No, it's not going to happen.
So I think this would be the last question for this session. We've had a lot of interesting questions.
Okay.
So there was a question which wanted you to comment about genetically modified organisms in the food industry or the synthetic industry that we have right now.
Well, that's just always that's ongoing. I mean, there's I think that does happen. A lot of it is because it's very interesting. I mean, it's a hard question for me to answer because I'm not a molecular biologist. I'm not a geneticist. But it does. Genetic modification of plants and things like that happens quite often. Mainly because what's the word? How the way to describe it? It's faster. It's faster to instill into that. I mean, you can't do that with humans because you've got a 90-year life of a human being, whereas a plant or a tomato, 1 or 2 years. So you can do modification a lot faster than you can with a human genome, which you're not allowed to. Anywhere on the planet, you're not supposed to be doing that.
Okay, thank you so much, Professor Amanda. I think it was really great, really a very great and insightful session. So I would like to invite Geetika Jain, who is my senior and the overall coordinator at Azeotropy, to present the concluding note.
Thank you. Thanks.
Hello, I hope I'm audible. Hello everyone, good afternoon. I'm Gitika and I'm overall coordinator for Azeotropy 2021. And I would firstly like to thank Professor Amanda Ellis for giving her valuable time to us and giving such an insightful session on DNA technology. I guess all the biotechs enthusiasts had a great time listening to you. And it was a great session for all of us. So thank you very much. And also, I would like to thank all the audience for attending this session. And yeah, that's it. Thank you so much.
Well,
Thank you
thank
so
you.
much,
It's been my
Professor.
thank you. Thank you for having me, and it's been my pleasure. And I wish you all the best for the rest of your conference. And maybe next year I could talk about something else.
Surely, yes, yes, Professor. Thank you so much.
Okay. Well, thank you very much. Thank you, everybody.
Bye bye.
Bye bye.